Hello everyone, welcome to today's topic, and today we will be understanding MHC class 1 molecule structure. So let me write down the topic, which is MHC class 1 major histocompatibility complex class 1 molecule structure. Very important structure, very important for T-cell activation, especially cytotoxic T-cell activation. When it comes to the structure, it has one heavy chain, which is alpha chain. So you can see three domains are there: blue, green, and the pink one. These are all the part of the alpha chain. So you have the first one, which is alpha 1 over here. Then you have the second one, which is alpha 2 right over here. And then you have third one, which is alpha 3. All three domains are really important for its structure. Okay, so I need to also create these NH2 groups, and there is the COOH group, which is right over here. So I'm going to create that so that you understand this is how the extension of this particular molecule is, right? And then you also have OH group, you also have phosphate group in the cytoplasmic tail region, right? And then let's talk about the parts. So you have cytoplasm is one section right over here. You have membrane protein, right? The section that is membrane, and then you have extracellular section. So this particular protein will have three sections: extracellular, membrane section, and then cytoplasm. This is the extracellular, and the next domain that we are discussing, beta 2 microglobulin. Beta 2 is very small. That is why it's known as microdomain, and it also has independent NH2 and COOH group, right? Okay. So the next is you also have disulfide linkage in these structures. Like in beta 2, you have disulfide linkage that is present, right? And further, you also have various other structures that are really important. Now let's talk about the protein or peptide binding region. So right over here, this is the antigen or protein or peptide binding region right over here, antigen binding site. So this is where it's going to have that peptide exposed, and then further cells, they're going to get activated with this interaction, right? Okay, now let's move on to the next part. Okay, so the peptides that are being exposed are of 8 to 10 amino acid long, so they are shorter than MHC class 2 peptides, right? And if you talk about alpha 3 domain is really important for CD8 cell activation. So basically, it's going to bind with CD8 co-receptor of cytotoxic T cells, and that is why it's going to play a very important role in activation. It also has CHO group, which is basically is a glycosylation. So it's really important for CHO. It's really important for T cell recognition. Next is you have transmembrane region. Let me label it right over here, transmembrane region, and then it's basically the alpha chain, coming from alpha chain only, not coming from the beta chain, right? So this is also known as cytoplasmic tail region. Okay, this is the cytoplasmic tail region, very short tail that is present. So you have CHO groups, and CHO group is added by the process of glycosylation, and that is really important for binding, okay, binding as well as binding with the CD8 receptor, and also for the stability of this entire structure, right? So these are some of the important components. Now let's see what are the important kind of peptides. So basically, intracellular peptides, they are expressed, or they are coming from viruses, tumor cells, and also intracellular pathogens. They are coming from these, and they will be exposed by these MHC class 1 and the cells, especially the cytotoxic T cells, in their activation process, right? So this is the entire structure of MHC class 1 molecule, and extremely important for antigen presentation, especially for cytotoxic T cells. Okay, I hope you enjoyed the video. If you like the video, then please hit the like button, subscribe to the channel, and support us. Thank you so much.